be the super fast-paced review of the D2 unit, starting with D2.1, cell and nuclear division. Before we even advance, let's get an overview of what we're about to learn. We're going to talk about mitosis and meiosis. These are both types of cell division, specifically division of the nucleus, but we're going to see that they have very different functions and they look very different. Okay, mitosis is going to take one diploid parent cell, okay, and it's going to produce two diploid daughter cells. Each one is genetically identical to the parent cell. Meiosis, on the other hand, is going to start with one diploid parent cell, and the end result is going to be four haploid cells, four haploid gametes. These are your egg and sperm cells that have half the genetic information so that they combine to form a diploid zygote. Note that in both of these processes, we start by replicating the DNA. And we're going to talk about how this is an important first step so that we have the right number of chromosomes in the resulting daughter cells. All right, let's go. Now, why do we need to do this? Mitosis is for growth, maintenance, right? If there's an injury, we would have to make more cells from the time you're born to the time you're an adult. Think about how much growth is happening, or even before birth. Okay, then reproduction, again, is where meiosis is going to come in to make those haploid gametes, egg and sperm cells. Okay, we call the cell that divides the mother cell or the parent cell, and the ones that are produced are the daughter cells. Okay, the mother cell disappears entirely as it's split into the two daughter cells. Okay, cell theory says that new cells are only produced by the division of pre-existing cells, and we can trace this back to the original cell, the zygote, that we all started as just one single cell. Okay, so let's talk about how the cytoplasm splits. This is called cytokinesis, and it's going to be different in animal and plant cells. So let's look at animal cells first. Okay, in animal cells, we're going to see the plasma membrane being pulled inwards around the equator of the new cell to form what's called a cleavage furrow. This is accomplished using a ring of these contractile proteins that are immediately inside the plasma membrane. The proteins are actin and myosin, similar to what we learned with muscle contraction. Okay, and when the cleavage furrow pinches in enough to reach the center, the cell gets pinched apart into two daughter cells. Fairly straightforward. Plant cells are going to be a little bit more complicated because plant cells have cell walls and a plasma membrane along the inside of that. So we're going to need to not just divide the plasma membrane and make new plasma membrane, but new cell wall. So first up, microtubules are built into a scaffold that straddles the equator, and that's used to assemble a layer of vesicles. Now we know that vesicles are made of phospholipid bilayer, which is what cell membranes are made of. And so those vesicles that assemble fuse together, okay, and form those two plates of membrane. So now we have division of the cytoplasm, but we still need to build that cell wall that's in between these two new cells. And so the next stage is that pectins and other substances are brought in by vesicles, and they're deposited by exocytosis between the two new membranes, and that forms that middle lamella, which is that space in between cells. Both daughter cells then bring cellulose to the equator, deposit it by exocytosis, and that is how the new cell wall forms. Okay, division of a cytoplasm is usually, but in not all cases, equal. So you can see here, usually goes right across the middle here where we see the new plasma membrane and cell wall forming. Okay, both daughter cells have to have at least one mitochondrion and any other organelle that can only be made by dividing a pre-existing structure. However, there's going to be a few exceptions, or we'll talk about two specifically, where instead cytoplasm is sometimes divided unequally. Our first example of this is budding in yeast. So yeast cells reproduce, okay, by first dividing their nucleus. Okay, the nucleus, one copy goes into this new bud that's forming. Okay, and a dividing wall is constructed, and this daughter yeast can split off and it leaves a scar. Okay, but this allows this process to occur repeatedly. Cells don't have to double in size between each division, and here you can see a micrograph of budding yeast. A second example is oogenesis in humans, which is egg production. Okay, Now we're going to see both sperm and egg cells are produced by the two divisions of a mother cell. In meiosis, there's two divisions. Okay, but usually we only produce one egg cell at a time. And this egg cell, though, is going to need a lot of food reserves because it has to support the developing zygote as it starts replicating until it's able to form a placenta once it reaches the uterus. And so we're going to see that in the first division, one large cell is going to form along with a small polar body, which does not develop further. And then after the second division, the same thing happens. Our second polar body doesn't develop further. This is a way to concentrate as much as we can, okay, of all of that cytoplasm filled with all of those organelles into the ovum so that it is as ready as possible to survive on its own if it gets fertilized. All right, now, if a cell was to divide without first replicating all of the DNA, okay, we would only have one cell that was able to get a nucleus if we didn't replicate, and the other one would be anucleate without a nucleus. Those cells wouldn't be able to synthesize polypeptides, okay? They wouldn't be able to grow or maintain themselves, and they would have very limited lifespans. Okay, and so instead what we're going to see is that replication occurs of all of the DNA. So we have two sets, and so when the cell splits, each resulting cell can get an entire copy of that DNA. All right, let's again compare mitosis and meiosis. Mitosis maintains the chromosome number. Each resulting daughter cell has the same number of chromosomes as the parent cell, the same genes, the same everything. They're exact identical copies. In meiosis, remember, we're going to have two cell divisions. We're going to start with the same thing, a diploid parent cell, okay? But we're going to have the genetic information so that each of those resulting gametes only have half as many chromosomes as the parent cell. And this is a random process of which half they receive. All right, so here we have a chromosome. A chromosome is just a length of DNA. Remember that DNA in eukaryotes is linear, okay? And we call one length a chromosome. Here you can see that this chromosome has condensed. It in this form would now be visible under a light microscope, okay? And when it starts to replicate, you're going to see that it's going to form what we call then a sister chromatid. This is an identical copy, okay? And when it splits, when mitosis or meiosis occurs, well, meiosis 2 specifically, we're going to see those sister chromatids pulled apart, okay? And we then call them chromosomes again. But we can see here these cohesin loops that are going to be split and cut in order for that splitting of sister chromatids to occur. Okay, so again, we're going to see that shortening and condensing of this DNA so that we can facilitate the division in half. Normally, DNA, if it's just in that tangled state, would be far too complex to try to divide it. And so this condensation allows for precise division to occur. Okay, I just said that. Okay, so how does that condensation happen? Well, we're going to see that chromosomes are initially condensed by wrapping the double helix of DNA around histone proteins, okay, to form these little packages called nucleosomes. Those nucleosomes are often referred to as beads on a string. Each DNA is wound around each set of 8 histone proteins 2 times, and then it's sealed with a linker protein, histone protein. Okay, so microtubules are going to be what organize the division of all of these chromosomes. Okay, we've got centrioles here on either end of the cell that are going to organize these microtubules and allow for splitting and division of those chromosomes towards each pole. Okay, this point where our two sister chromatids come together is called the centromere, and on the outside of the centromere are kinetochores. And we're going to see the kinetochores act like little motors that are going to shorten these microtubules, therefore pulling the sister chromatid, in this case, towards the pole. Okay, so how does that work? Okay, the kinetochore, again, is like a little motor, and it shortens the microtubule by removing these little tubulin proteins. And so by removing them, one by one, it shortens it and it allows for pulling of those chromosomes. Okay, so we're going to see in meiosis, we're going to have alignment of homologous chromosomes, and each, you can see, are made up of two sister chromatids. Okay, but these are like the two you got from mom, and well, the one you got from mom that's been replicated and the one you got from dad that's been replicated. Those homologous chromosomes are going to be joined together at the chiasmata, which is also, we're going to see the point where crossing over can occur. All right, let's run through the phases of mitosis. So first up, this is even before mitosis starts, we're going to see that cells for most of their life is in the phase interphase. Interphase, we've got all of our DNAs decondensed. This is where the cell is doing its job, right? It's using that DNA as a code to make proteins. It's very metabolically active. It's doing cell respiration. Okay, and then at one point in the middle of interphase, we're going to see that the cell replicates all of its DNA. So by the end of interphase, it's going to have 2 copies of DNA. Now, you should note that you should be able to recognize what these different phases look like in a micrograph. And so if you see a cell where it just has a whole bunch of DNA that isn't in clear chromosomes, it's not been condensed, then you know that that cell is in interphase. All right, so now we're going to start mitosis. Mitosis is made of 4 stages. The first stage is called prophase, and that's easy to remember because pro means before, and it sounds kind of like pre, like something that happens first. And so we're going to see many things happen in prophase. I'll look here, we can see our chromosomes are going to condense down. They're now visible. Okay, we're also going to see microtubules start to grow from these structures at the poles of the cell called microtubule organizing centers. They form this spindle-shaped array. Okay, and then at the end of prophase, we're going to see that the nuclear membrane breaks down. Now we're going to move to step 2. Step 2 is metaphase. I like to think of it as meat in the middle, all of those M's. Here we're going to see our pairs of sister chromatids line up along the equator right in the middle of the cell. Okay, microtubules are going to attach to those centromeres. Okay, the spindle microtubules are put under tension to test whether the attachment is correct. Okay, and by the end of metaphase, they're all lined up in the middle. Next up, we have anaphase. Anaphase starts with an A, just like the word away. This is where we now have splitting of those sister chromatids, and we call them chromosomes again. Okay, the kinetochores are shortening these microtubules, okay, pulling each of the copies of that DNA to its own pole so that each of these resulting what will eventually be cells have the exact copy because these are copies of the same chromosomes that are being split. So there's going to be identical cells produced at the end. Okay, and then lastly, we have telophase. Telophase is where we're going to have our nuclear membranes reform. Okay, our chromosomes are going to decondense again. Okay, and cytoplasm is usually being split through cytokinesis, which we talked about earlier, already by the time that telophase is complete. So we have now created 2 separate cells from one. Each cell, okay, is diploid, just like the parent cell was, and this is how, again, our bodies grow. Our bodies replace cell tissue. Now, you should be able to recognize each of these different phases on a micrograph. So you can see here, most of these cells are in interphase where you cannot see distinct chromosomes. Here we have an example of prophase, okay, with condensed chromosomes. Here we have an example of metaphase where they're meeting at the middle. So is this one here. Okay, here we have an example of anaphase where the chromosomes are moving away from each other. And lastly, here you can see telophase. These 2 small cells, we can see cytokinesis has begun. You can see, though, that those chromosomes are decondensing and our 2 new cells are almost split. Now let's shift gears and look at meiosis. Now, we have what are called homologous chromosomes. This is one you got from dad and one you got from mom. Now, these are the same chromosome number. Remember, we talk about how we have 23 pairs of chromosomes. So say, for example, this could be Chromosome 7. You have a chromosome 7 from dad and a chromosome 7 from mom. They have the same genes at the same loci, but they might have different forms of the gene called alleles. Okay, and these alleles can be reshuffled during meiosis, and we call that recombination. Okay, so we're going to see that we start with the same first step during interphase. Okay, the DNA is going to need to replicate. And so each of these chromosomes that are homologous have a sister chromatid. Remember that sister chromatids are just the identical copy. Okay, now your body cells, okay, are all going to have one of each, or two of each chromosome, one from mom, one from dad, and we call that 2N or diploid. Okay, sex cells, though, we're going to see at the end of the process of meiosis, which creates sex cells or gametes, that we're going to have haploid cells, which we indicate by N, and they only have a single set of chromosomes, all of which are non-homologous. Okay, that way, when two haploid cells join together at fertilization, okay, we can form a diploid cell that has one chromosome from each parent of each pair of each type. Okay, meiosis is the reduction division. Okay, again, this happens during the process of gametogenesis or genesis, formation of gametes. If we didn't divide the chromosomes in half, then sexual reproduction would result in tetraploid offspring where they would have 4 of each type of chromosome. So let's look at how we can split the chromosomes in half. We're going to see that we run through those same 4 steps: prophase, metaphase, anaphase, telophase, 2 times. We have 2 divisions in meiosis. Now, the second division we'll run through looks exactly like mitosis. This is where we have separation of sister chromatids. Okay, but in meiosis, something special happens. We're going to see that the homologous pairs are going to be what line up and get separated during this first round of division. Okay, so, and then the second round is where we're going to split the sister chromatids. Okay, again, at the end of meiosis, we're going to end up with 4 haploid cells. Okay, our homologous chromosomes are separating each other in anaphase 1, and then those chromatids separate in anaphase 2. Okay, but sometimes errors occur. Here you can see that both of these homologous chromosomes have moved to one pole. We call this non-disjunction, and in this case we know it's in anaphase 1 because these are homologous pairs that fail to separate. But this can also occur in anaphase 2 because here you can see there's sister chromatids that failed to separate. Okay, now most of the time, okay, a missing chromosome is going to lead to death of the cell. Having an extra chromosome is also in most cases lethal. Okay, however, we know that there are exceptions to that. In a few cases, it's not lethal. For example, Down syndrome is due to non-disjunction, okay, where that person has 3 copies instead of 2 of chromosome number 21. Okay, and so here we can see non-disjunction happening. Okay, so we have then both chromosomes went to one gamete, and so when that gamete combined with another gamete, okay, we end up with 3 instead of the usual 2. Okay, now note this can also happen with the sex cells. You don't need to know details, but just be aware, okay, that there are some syndromes here where non-disjunction occurs, and you can have either just a single X chromosome or 2 X chromosomes and a Y. All right, meiosis generates genetic diversity in 2 ways. Either random orientation of the bivalents, which are paired homologous chromosomes, okay, or through crossing over, which we'll cover. Okay, so let's just start before we even jump into these. Random orientation refers to which side of the equator each of those homologous paired chromosomes line up on. You can see that there's just going to be a 50-50 chance. Say that these red chromosomes were all the ones that came from mom, and these were all the green ones from dad. Okay, so in this case, we're seeing different combinations of how they could set up as they line up in prophase 1. And so this random orientation is going to lead to a ton of genetic diversity because when these homologous chromosomes are split, the 2 resulting cells each are going to be different from each other. Okay, now this is only a set of 3 paired chromosomes. Remember that humans have 23 pairs of chromosomes. And so we're going to have a huge amount of genetic variability from that random orientation. Okay, what about crossing over? Now, crossing over occurs during prophase 1, okay, when our homologous chromosomes pair up next to each other in preparation to be split, okay, and this is called a bivalent. When it's paired, that's called synapsis, okay, but we can see 2 non-sister chromatids, okay, exchanging genetic information, okay. Crossing over can occur at any position along the chromosome. At least one crossover occurs in each bivalent. There's often more than one. Okay, so they're exchanging the same genes, but just again, there can be different alleles, different forms of the gene. Okay, here's another example, just touching on random orientation, but I explained that a couple slides ago. All right, so now we're going to run through the steps. We'll begin with the first division of meiosis. Again, we're going to run through prophase, metaphase, anaphase, telophase 2 times. So first up, prophase 1, synapsis occurs. Our homologous chromosomes form parallel pairs. Crossing over occurs during prophase 1 between non-sister chromatids. Okay, and then of course those chromatids shorten and thicken and they condense, so they're now visible under a light microscope. Next is metaphase 1, meet at the middle. Our homologous chromosomes line up along the equator. Okay, and we're going to see now that in anaphase 1, they move away from each other. So these paired homologous chromosomes separate. You can see here that crossing over has occurred in both bivalents. This again is driven by those kinetochores that are shortening those microtubules. Okay, the chiasmata slide to the end of chromosomes. The chiasmata, remember, are where those homologous chromosomes were joined. Okay, and then we wrap up the first division with telophase 1. Just like before, we're going to see that the nuclear membrane is going to reform. Cytokinesis divides the cytoplasm, and the chromosomes are going to decondense. But now it's time for the second round. So whereas we just split up homologous pairs, the second division is going to look just like mitosis in that now we're going to split sister chromatids. So in prophase 2, same thing happened. We condense our chromatids, our nuclear membrane breaks down, okay? And we are then going to move into metaphase 2. This is where they meet in the middle, okay? The cohesin loops get cut. And then in anaphase 2, the kinetochores start pulling and separating those sister chromatids towards each pole. And at the end, okay, we are left with telophase 2. We have reformation of the nuclear membrane. Our DNA is going to decondense. Cytokinesis occurs, and we now have a total of 4 daughter cells. Each one has half of the genetic information as the parent did. All right, this is the end of SL content for D2.1. Let's continue with HL content. All right, cell proliferation is a rapid increase in the number of cells. This happens when cell division happens at a faster rate than cell death, and this is needed for growth, cell replacement, and tissue repair. Okay, so in most animals, we have embryonic and juvenile phases of growth, after which adult size is reached, growth usually stops. Okay, in some parts of the body, those cells continue to proliferate during juvenile phases of growth. For example, growth plates near the end of bones are active in childhood and adolescence. Cell divisions contribute to bone growth and rapid increase in height. Now, in plants, we have growth regions called meristems. Okay, here's an apical meristem. It's found at the tip of stems and roots. Okay, some of those daughter cells are going to just continue to divide in these locations. Okay, and the cells that are produced will differentiate for a specific function. Okay, we know that cells have to be replaced. Here's an example of skin cells, okay, that are constantly being made because the outer layer of your skin is made of these dead cells that eventually are going to slough off, and so they're constantly being replaced through the process of mitosis. Okay, and then tissue repair. If there's a wound to a part of the body, Okay, we can heal it by regenerating new cells through mitosis. All right, this cell cycle. So this is the cell's life cycle. Most of the time is going to be spent during interphase. Now, don't make the mistake of thinking interphase is boring and nothing's happening. This is a highly metabolically active phase. This is where the cell is doing its job, right? If it's a neuron, it's sending electrical signals. If it's a cell that's making a hormone, right? It's producing all of those hormones. It's doing its job. Okay, it's growing. But at this second phase of interphase, the second part is called the S phase or synthesis phase. And this is where DNA is replicated. Okay, so then we're going to have our growth, replicate our DNA, grow a little bit more, and then we're going to get to mitosis. Mitosis is nuclear division. Here's those 4 steps: prophase, metaphase, anaphase, telophase. Okay, technically mitosis is nuclear division, so we say that after mitosis, then cytokinesis occurs. Okay, that's splitting of the cytoplasm, and then we are back around to interphase again. All right, if you remember from when we looked at cell organelles, okay, this we can recognize as a nucleus because remember it has all of these sort of dark splotches. So let's identify now what those are. Okay, those dark areas are called heterochromatin, and that is where the DNA is really, really tightly compacted because these are going to be the parts of the DNA that are not used by the cell to make proteins. Because remember, only the parts of the gene that codes for proteins that are relevant to that cell's job are going to need to be protein synthesized through transcription and translation. Okay, on the other hand, chromatin, or sometimes referred to as euchromatin, is going to be the more decondensed part of the DNA that actually goes through transcription and translation to make proteins. Okay, note cells following the cell cycle normally double in size during interphase. Okay, DNA gets doubled by replication, the volume of the cytoplasm increases, and the area of the membrane is increased. Okay, the numbers of most types of organelle have to be increased. Now, mitochondria and chloroplasts contain DNA, so they can do self-division. Other membrane-bound organelles, such as the Golgi bodies, bud off from existing ones. Non-membrane-bound organelles are assembled, like ribosomes. Ribosomes are assembled in a region of the nucleus called the nucleolus, which is this dark spot here. Okay, remember, interphase is an active phase in the life of most cells. Many metabolic reactions are occurring. Okay, biosynthesis of protein and other molecules. The cell is just busy doing its job. Now, how do we control when the cell moves to the next stage of the cell cycle? That's going to happen using cyclins. Now, cyclins are a group of proteins that start to build up, and then when they reach a certain threshold, that signals the cell that it's time to move on to the next phase. Here you can see some different cyclins. You do not have to memorize any details about which cyclin does what. Just know generally that as they build up and reach a threshold, that triggers the cell to move to the next phase in the cell cycle. Now, sometimes there are mutations in the genes that control the cell cycle. Okay, if we have problems and the cell starts dividing out of control, that's what we call a tumor. And there are mutagens that can increase the chance of tumor formation. Okay, mutagenic chemicals. We have over 50 substances that are definitely carcinogenic or cancer-causing. Okay, all forms of high-energy radiation such as X-rays and ultraviolet rays, but not visible light or other forms of the lower energy radiation. Okay, there's 2 groups of genes that can change a normal cell into a tumor if it mutates. First up, proto-oncogenes. Know that onco is referring to cancer. The cancer unit at a hospital is called the oncology unit. So proto-oncogenes. Okay, some of these regulate expression of genes concerned with cell proliferation. Okay, others have roles in secondary messenger systems that control the cell cycle. And a third group is concerned with growth factors. Now, if these mutate into oncogenes, they can actively promote cell proliferation. They're also genetically dominant. So only one of a pair of proto-oncogenes has to mutate for that oncogene to be formed, which then increases the chance of a tumor. Okay, next we can have tumor suppressor genes. These prevent cell proliferation. Some of them function as brakes at checkpoints. Others are needed for DNA repair to correct errors in replication. A third group has roles in programmed cell death. And so if we have mutations to these tumor suppressor genes, that can then increase the risk of tumor formation if it's no longer functioning properly. Okay, any cell in the body can become a tumor cell, but normally one mutation isn't going to cause a tumor to form. Usually there's going to be more, as many as 10. And the chance of this happening is very small, but humans have long lifetimes, and so mutations are possible at any time. We also have a large amount of cells in our body, and so that's why sometimes these errors do occur. Okay, when a tumor has formed, we call it the primary tumor. Okay, and if the cells are all sticking to each other, okay, and they're not spreading, then that's a benign tumor, okay, and it's not thought of as cancer. But in other cases, the cell-to-cell adhesion is not adequate. The cells may invade neighboring tissues. We call that spread of tumor cells from one part of the body to another metastasis. Okay, and if that occurs and those spreading cells end up forming secondary tumors, then we call those malignant tumors, and that's commonly known as cancer. Certain tissues are more likely to produce those malignant tumors, like places in the breast, ovaries, testes, and the thyroid gland where there is hormonal stimulation of cell division. Okay, here's just an interesting MRI scan. Here's a patient without cancer, and then this is a patient where you can see secondary tumors have developed in the brain following breast cancer, so metastasis has occurred. Okay, now a diagnosis of cancer is always worrying, but there are effective treatment options that have been developed for many types of tumor. Okay, the rate of cell growth and division in tumors is variable, and in some cases can be very slow. Okay, the last skill for this subtopic is you should be able to calculate a mitotic index, which is just a ratio of the number of cells in mitosis to the total number of cells. Okay, so if you were given this image, you would need to count up the number of cells in any of the 4 mitosis stages: prophase, metaphase, anaphase, or telophase, okay, divided by the total number of cells, and that gives you the mitotic index. Okay, on to D2.2 gene expression. Now, this is an all HL subtopic, so SL students don't need to tune back in until we get to D2.3. All right, first up, the genotype of an organism is all of its genetic information, every single bit of DNA in a cell that has all of your DNA. Okay, and the genotype codes for the phenotype. pH, just like a physical expression, this is then what are the physical characteristics, the structures and functions that are coded for by the genotype. So gene expression is the process of turning the genotype into the phenotype. Okay, so this is just a quick reminder. Remember that transcription occurs in the nucleus as a DNA is made a copy of a portion of it that's called a gene into mRNA. That mRNA then exits the nucleus, and a ribosome then translates it into a growing amino acid chain called a polypeptide, which then folds into a protein. Okay, protein can form part of the structure of an organism, but many have other functions, so their effects are not necessarily visible in the outside appearance of an organism, but the effects are still observable and are therefore part of the phenotype. They could be enzymes, and so their effect on the phenotype could be by catalyzing a reaction. Now, gene expression is not necessarily just an on or off process. It also can be assessed by measuring the quantities of proteins or other gene products. All right, so let's talk about some ways that we use regulation of transcription by proteins that bind to specific base sequences in DNA. Okay, so we're going to have selective transcription of that RNA. Okay, the enzyme RNA polymerase is going to bind to a sequence close to the gene known as the promoter. Okay, so you can see it's just upstream of the gene that we may or may not be turning into mRNA. Okay, so a common feature in eukaryotes is the TATA box, a short sequence of bases that is part of the promoter. We're going to see a generalized transcription factor bind to it, and this is going to indicate to RNA polymerase where transcription should be initiated. Okay, every gene in an organism has a similar promoter, okay, but only some genes are going to be selected for transcription at any stage in the life of a cell. So selectivity of that transcription is based on DNA binding proteins. These can locate and reversibly attach to specific DNA base sequences. Now note that this double helix has what is called the major groove and the minor groove. Okay, the major groove is wider, the minor groove is narrower. Okay, and in most cases, those DNA binding proteins are going to bind to a major groove, okay, as shown in these two images. Okay, but sometimes it can go bind to a minor groove. Now, the DNA binding proteins that regulate transcription are called transcription factors. Their binding sites lie upstream of the gene. The base sequences of the binding sites are very variable, okay, in some cases unique, which allows for regulation of single genes or specific groups of genes. Okay, in contrast, the promoter sequence where the RNA polymerase binds, okay, is shared. So transcription factors can act as activators or repressors, and they can turn genes on and off, okay, by binding to these regulatory sequences on the DNA called enhancers and silencers. Enhancers are sequences where activators bind and increase the rate of transcription. Silencers are sequences where repressors bind and decrease the rate of transcription. Okay, so we just have proteins called transcription factors binding to sequences, okay, which either encourage, okay, or silence the transcription of the gene that is downstream. Okay, we can also regulate gene expression after transcription occurs at a number of levels. One mechanism is control of the degradation, or how fast it takes for that mRNA to break down. So after transcription is complete, okay, a poly A tail is added to the end of the mRNA strand. This increases the stability, but it does get shortened over time. And the shorter the poly A tail, the less likely the mRNA is going to be translated, and the more likely it's going to be degraded by a nuclease enzyme. Nucleases are going to break down nucleic acids. So the rate of shortening of this tail varies by a factor of over 1,000. In some types of mRNA, 30 nucleotides can be removed per minute. In others, only 1 or 2 per hour are removed. So given an average tail length of 200 nucleotides, this implies that mRNA molecules might persist for between 5 minutes and a whole week. So depending on how long it persists, that's what's going to affect how much protein is made from the mRNA's code. Okay, some proteins are produced briefly, such as those that control the cell cycle. Okay, others, such as the milk protein casein, must be produced continuously for long periods of time. Okay, if we have, for example, a lactating mother. This necessitates regulation of mRNA degradation. Okay, hormones and other chemicals can also influence mRNA degradation rates. Okay, so you don't need to know much detail about that, but just know that different hormones like estrogen, okay, can affect the rate of transcription of a particular protein. Okay, epigenesis is the concept that a multicellular organism, such as a plant or animal, develops from undifferentiated cells. Structures and functions then appear during development that were not present at the start of an organism's life. How does this differentiation of cells occur? Okay, well, we activate certain genes and we silence others. Remember, every gene is in every cell, so it just depends which ones we actually end up turning into proteins. Now, this can also be controlled by chemical modifications to DNA and the proteins associated with DNA that determine if it's going to be activated or deactivated. These modifications are known as epigenetic tags. Okay, they're going to influence the genotype, or they don't influence the genotype of an organism, right? We're not changing the bases and their order in any way. Instead, we're just adjusting the phenotype, okay, as it controls which actual genes get turned into proteins. DNA-based sequences, again, are not altered, okay? But we're going to see these epigenetic tags affecting transcription. Okay, a few words you need to know. The genome is the whole of the genetic information of a cell, including coding and non-coding sequences, all of your DNA. The transcriptome, okay, is the entire set of mRNAs transcribed in a cell, and no cell is going to transcribe every single gene that is available to it. So the transcriptome is going to be less than an RNA copy of each protein coding gene. It's going to vary over time as well, okay, depending on the cell's needs and between cells within an organism. Okay, the proteome is the entire set of proteins that can be produced by a cell. It's based on the transcriptome because proteins are synthesized from mRNA, but the quantities of each protein in a cell are not directly proportional to the quantity of the corresponding mRNAs. Okay, we're going to see this as part of the control of gene expression. That's going to determine how that cell differentiates. All right, so let's get down into epigenetic tags. Methylation of DNA is where we take a hydrogen and we substitute it with a methyl group. Okay, this most frequently occurs on cytosine, the C base, okay, but it sometimes can occur on A adenine. Now, the pattern of methylation where these methyl groups are added, okay, changes during the lifetime of a cell, and it's affected by environmental factors. Again, it doesn't alter the genome. The sequence of bases remains the same. Okay, and the reason it doesn't alter is because the methyl group is in a position where it doesn't affect complementary base pairing. So that whole process stays the same. Okay, this is an example of an epigenetic tag, and methyl groups have multiple roles in the regulation of gene expression. So remember, the promoter here is a base sequence that's located near a gene. It's the binding site for RNA polymerase. If we methylate cytosine in the promoter region, that is going to tend to repress transcription of the gene and therefore its expression downstream. So the more methylation that we see in the promoter region, the less chance that this gene is going to be transcribed. And methylation, okay, is something that occurs based on environmental factors. Okay, DNA is combined with histone proteins. We already talked about this to form these nucleosomes. We've got 8 histone proteins. The histones have these long tails. You can see here, they extend out of this compact nucleosome. Now, these tails can interact with neighboring nucleosomes, resulting in tight bonding during condensation of chromosomes. So these histones play a role in a tight packing of DNA. They also play a role, though, in regulation of gene expression. Different types of modification can occur to the amino acids, okay, in the tails of histones, including the addition of methyl, acetyl, or phosphate groups. These are also all examples of epigenetic tags. Methylation of amino acids in histone tails can influence transcription of a gene by either decreasing or increasing the accessibility to transcription factors. You don't need to know any details of how that is achieved. Okay, chemical modifications of chromatin, such as methylation of bases in DNA and acetylation or methylation of amino acids in the tails of histones, are called epigenetic tags. They have an impact on gene expression. They affect the visible characteristics of an individual. The sum of all the epigenetic tags in a cell or organism is called the epigenome. Now, each type of cell in a multicellular organism has its own distinct patterns of epigenetic tags, so it produces the proteins needed to perform its functions. Now, when cells in a tissue divide by mitosis, one parent cell becomes 2 identical daughter cells. The epigenetic pattern is passed on to the daughter cells. As a result, okay, the new cells are differentiated for the same functions. Any effects of the environment on the epigenome are conserved. In meiosis, where we form haploid gametes, okay, they have a characteristic pattern of epigenetic tags. Some of the pattern is passed on to the haploid cells, okay, but it has been assumed that the epigenome will be entirely erased, okay, before gametes fuse to produce a zygote. Okay, the process of wiping the epigenome is called reprogramming. However, there is now evidence that a few epigenetic tags may not be removed. They would remain in the zygote and thus are passed from parent to offspring, which means that there can be environmental influences that then get passed on. This is known as transgenerational epigenetic inheritance. This is being highly researched right now. It opens up the possibility that the environment encountered by one generation could have an impact on gene expression in the next generation. Okay, it should be stressed this doesn't amount to the environment directing specific inheritable changes in genes. Okay, so we're not Lamarck here. Even if an epigenetic tag is passed from parent to offspring, it can easily be reversed, unlike mutations in DNA, which are changes to the base sequence and are likely to be permanent. All right, other examples of environmental effects. Air pollution directly damages tissue, but there is also strong evidence that it can alter the epigenome. Exposure to air pollution, has been found to decrease DNA methylation across the genome in people of all ages. One area of impact is in the expression of proteins that regulate the immune system. These changes could be the cause of increased risk of heart disease and evidence of inflammation and diseases such as asthma. Air pollution is particularly damaging during pregnancy because it causes changes to methylation in specific genes that have impacts both during gestation and the early years of life. All right, so when sperm and eggs are produced, approximately 99% of epigenetic tags are removed. But some of those tags, again, that are acquired from the environment of the parent, persist into the next generation. This is non-Mendelian inheritance. It's sometimes known as genomic imprinting. And then that can be maintained because epigenetic tags stick around through mitosis as that zygote replicates and differentiates and grows. And so in this case, we're going to see that we can have the allele from a gene inherited from one parent be silenced, the allele from the other expressed. So even though two copies of the gene are present, it's as if the individual were haploid. Alleles and mutations that might normally be recessive might be expressed if the dominant allele is silenced by imprinting. So for example, if we had a mother who is homozygous recessive for a condition and the father is homozygous dominant, genotypically, the offspring would be all heterozygous, and we would expect it to have the phenotype associated with the dominant allele. However, if the allele inherited from the father is silenced because of these epigenetic tags, we might see that the recessive condition ends up appearing in the offspring. One condition that is caused in this way is called Angelman syndrome. You don't need to know any details about that. Lions and tigers are closely related, so they have been known to interbreed in zoos. The outcome of the cross differs depending on whether the mother or father is a lion. Now, female lions carry litters from multiple fathers, so there's an advantage to the father to sire the largest sized cub of the litter. There is an advantage to the mother to sire the largest number of cubs. Paternal imprinting favors the production of larger offspring, and maternal imprinting favors smaller offspring. Now, normally these competing priorities balance out when both parents are lions. Tiger litters, on the other hand, are sired by a single father, so there's no competition that favors imprinting. So consequently, if we have a male lion that breeds with a female tiger, we're going to see that the offspring tend to be larger than either of the parents, whereas if the male is a tiger, the offspring tends to be smaller than either of the parents. All right, there are two types of twins. We can have two different eggs released from the ovaries. Each egg gets fertilized by a different sperm. And these are called dizygotic or fraternal twins because there's two different zygotes. They're only as genetically similar as any other two siblings that share the same mother and the same father. About 50% of their genetic information is in common. On the other hand, about 1 in 250 pregnancies results in the formation of monozygotic or identical twins. This occurs where we have one fertilized egg, but very early in development, it splits and each of those forms an individual, and they're therefore going to have the same DNA. And so we can do studies with these monozygotic or identical twins to see how the environment has affected them over their lifetime. So here we have a study that compared methylation patterns of 3-year-old identical twins and 50-year-old identical twins. Methylation patterns were dyed red on one chromosome for one twin and green on the other. Okay, we can see that the 3-year-old twins have very few differences. There's very few methyl groups that are different from each other. There's a lot of yellow, right, which is the same as the other one. Note that by the time this set of twins got to be 50, there is a ton of red and green, indicating methylation that's on one twin and not on the other twin. And so we can see that over this 47 years, right, now these are different sets of twins, but this is going to be consistent with older twins are going to have more differences because they have different environmental factors that have been acting upon their lives, right? They're eating different foods, they're exposed to different environmental toxins, okay? All of the things that we go through every day in our lives. Okay, variations in environmental factors can have an impact on gene expression. For example, the genes responsible for the absorption and metabolism of lactose, okay, in the bacterium E. coli, they're expressed when lactose is present and not expressed when lactose is absent, right? We don't want to make lactase that breaks down lactose if there's no lactose sugar present to break down. That would be wasteful. And so we're going to see the breakdown of lactose results in regulation of gene expression by negative feedback, okay? And so we're going to have a repressor protein that gets deactivated when lactose is present, and so it's no longer going to repress the formation of lactase, and so lactase is going to be able to be produced. Once the lactose has been broken down, though, the repressor protein no longer is deactivated, okay, and so it's once again going to block the formation of lactase by blocking the transcription of the gene that codes for lactase. Okay, and finally, just know that hormones can also affect gene expression. Here we can see estrogen binding to an estrogen receptor. Okay, this then can bind to that promoter region, okay, and allow for a gene to be expressed. All right, let's move on to D2.3, our final slideshow for this unit. Okay, water potential. So first up, solvation is the combination of a solvent with the molecules or ions of a solute. Okay, we're going to see the properties of water as a solvent depend on water's polarity. We have a partial negative charge, we know, at the oxygen end and partial positive charges at the hydrogen poles. Okay, we're going to see that polar solutes dissolve due to the attraction between partial positive and negative charges on water molecules. We're also going to see positively charged and negatively charged ions. Okay, here you can see our salt crystal, okay, NaCl. The Na sodium is a positively charged ion, and so we're going to see that when you add salt to water, it's going to dissolve because we're going to have all of these partial negative oxygens attracted to the positive sodium ion, and then vice versa, we're going to see all these partial negative hydrogens attracted, partial positive hydrogens attracted to the negative chloride ion. Okay, here you can see that again. Then note, we also have all of these water molecules that are attracted to glucose because glucose is polar. Okay, it has polar hydroxyl groups. Oxygen has a partial negative charge, hydrogen a partial positive charge. So we're going to see hydrogen bonds formed between these hydroxyl groups and water, which is also why then sugar dissolves in a glass. All right, the particles in a liquid can move, but because of intermolecular attractions, they don't separate from each other completely unless the liquid is changing to a gas. Hydrogen bonds are being repeatedly broken and formed in water. Okay, many bonds exist at any given time, so there's a strong overall attraction between water molecules. Let's see. All right, so we're going to look back at osmosis, which this should be review. Remember, osmosis is the net movement of water from a less concentrated to a more concentrated solution across the membrane. Okay, so if here we have a semi-permeable membrane, this is less concentrated, this is more concentrated. If the solutes are too large to move across the membrane through diffusion, we're going to see movement of water only, and that's called osmosis. And we know that we want both sides to even out their concentrations. So water is going to go from a low solute to a high solute side of the membrane until it's at equilibrium. Now there's still going to be movement of water molecules, but there's no net movement at this point because the concentrations are balanced out on either side. All right, now we are going to use the term hypertonic solution to talk about a solution with a higher solute concentration. Okay, the word hyper is higher, okay, like hyperactive. A solution with lower is going to be hypo, like a hypodermic needle goes under or below the skin, the dermis. Hypotonic has a lower solute concentration. So we're going to see that there's a net movement of water from a hypotonic solution to a hypertonic solution. Now, eventually, okay, those solutions are going to become isotonic, which means the same. Okay, there's no net movement of water at this point once the concentrations are the same. Okay, when we say concentration, we're talking about the amount of solute per unit volume of solution. The volume can be measured in cubic meters or decimeters. Liters are still sometimes used as volume. Negative indices indicate per unit volume. So if you see a negative in a exponent, that's just the same as it being in the denominator, right? So we would read this as like per cubic meter. Okay, the amount of solute is measured in moles. So a sodium chloride solution has a concentration of 0.5 moles per cubic decimeter. All right, all living cells have a plasma membrane. Remember, even plant cells, even though they have cell walls, there's a plasma membrane that controls what comes in and out of the cell that's just inside of that cell wall. Okay, the membrane of most cells is very permeable to water, but much less permeable to solutes. So we're going to see net movements of water across the membrane through osmosis, which is a passive process. Okay, we call plasma membranes semi-permeable because they're not freely permeable to all particles. Okay, so osmosis is passive. Let's see, cells can change how rapidly osmosis occurs by changing the permeability to water of their plasma membrane. They can change direction of movement by raising or lowering the concentration of solutes, okay, because water always moves from hypotonic to hypertonic solutions. Okay, root cells absorb water from the soil because the cytoplasm of cells is hypertonic compared with water in the soil. All right, I'm skipping a practice problem. So let's look what happens. Animal cells have a plasma membrane but no cell wall. Let's compare plasma membranes of plants and animals to cell walls of plants, which again have a plasma membrane lining them. Plasma membranes of animal cells are made of phospholipids. They're thin, they're very liquid, they're very flexible, low tensile strength, semi-permeable. Cell walls of plants are made of cellulose and they're much thicker. They're extremely solid, they're not moving around, high tensile strength, okay, but they're freely permeable to materials through all of the gaps and openings. So if an animal cell is bathed in a hypotonic solution, okay, with a lower concentration, that means water, we know, always is going to go from low to high solute concentration. So water is going to enter the cells, okay, making them swell up, okay, and potentially burst. On the other hand, if an animal cell is bathed in a hypertonic solution, which is highly concentrated, okay, then we know water is going to move again from low to high solute concentration, and the water is going to be exiting the cell, and those cells can shrivel. Isotonic, remember, is where cells are in the same concentration as the cells themselves, and so we have no net movement of water. Okay, you should be able to recognize images of red blood cells to determine what type of solution they've been placed in. Okay, so note this is normal. These are those typical red blood cell shape. They're in an isotonic solution of the same concentration. Okay, these are shriveled red blood cells. They've been placed in a hypertonic solution. And these are swollen. You can see that they've blown up. They're spherical almost. So that's in a hypotonic solution. Okay, in unicellular eukaryotes like this protist paramecium Aurelia, we're going to note some contractile vacuoles. That's going to allow for fast movement of water. All right, what about when we have plant cells? Now, Plant cells can withstand high pressure when water moves in because of how strong their cell walls are. If water is moving in because this plant cell is in a hypotonic solution, we're going to see high turgidity. It's going to have lots of pressure of that water moving in. Okay, this is the normal state of plant cells. This is what helps plants stay upright and not wilt. If a plant cell was in an isotonic condition, we're going to see, again, no net movement of water. Okay, but you can see that's fairly flaccid, and that's not going to allow for a plant that doesn't have like woody tissues to stay upright. Picture like a green plant that you might have on your windowsill. Okay, if they're in a hypertonic solution, we have what's called plasmolysis that can occur, where so much water leaves the cell, okay, that the plasma membrane actually pulls away from the cell walls, okay, and we call that then a plasmized cell. Okay, we covered all of this, and we covered all of that. All right, this is again just review. But let's look at then an application. If this was a medical procedure, we'd want to be sure that we would use a normal saline solution, which has the same concentration as the cells in your body. So let's look at a couple of medical applications of isotonic solutions. Okay, so if you're ever given an IV for rehydration, for example, that's going to be using that saline solution. We don't want to put just pure water into your blood, right? Or think about the resulting effects on your red blood cells. Okay, saline then can also be used to rinse wounds and skin abrasions for the same reason. It can be used to keep areas of damaged skin moistened prior to skin grafts. And it's used as a basis for eye drops. Okay, lastly, it can be used in a slushy form to package donor organs for transport between hospitals. All right, that takes us to the end of SL content for D2. SL students, you're good to go. HL students, we have a bit more. All right, now let's talk about water potential. So remember potential energy from a physics class you might have taken. Okay, this ball has potential energy because if it was released, not even thrown, just dropped, it would drop, okay, due to the potential energy from gravity. We're going to now talk about the movement of water as the water potential, okay? And it's going to have a couple of factors that are going to go into play. When we talk about water potential, you're going to see this symbol, the Greek letter psi. It looks like a little trident, okay? And it's usually measured in kilopascals or sometimes megapascals. Okay, now we can't measure the absolute quantity of potential energy of water, so all values are going to be relative to this standard we've set: pure water, standard atmospheric pressure, and 20 degrees Celsius. That's been assigned a water potential of 0, and everything that we're going to talk about now is in comparison to that standard. All right, many factors can influence water potential, but in living systems, we only care about 2 different factors: solute concentrations and hydrostatic pressure. Rises or falls in hydrostatic pressure can change the potential energy of water. The higher the pressure, the more potential energy water has. Okay, and when solutes dissolve, the potential energy of water is reduced. The higher the solute concentration, the less potential energy the water has. Now, water potential allows us to predict the direction in which there's going to be net movement of water molecules. This is what you need to memorize. Water always moves from a higher to lower water potential because this minimizes potential energy. Just picture, okay, like a rock rolling down a hill, right? Or going back to this girl, okay, we're always going to go from higher to lower, okay? A rock that has potential energy at the top of the hill is going to roll down the hill, higher to lower. So same thing here. Water goes from higher to lower water potential to minimize its potential energy, okay? And so we're going to see the range of water potentials, it's always going to be either 0 or a negative value. And so if you were asked here, which way would water go? We know that water is always going to go to a more negative value. So water would move, from this case, from left to right because of negative values, right? Negative 300 is lower than negative 200. All right, so solute potential, okay, is indicated with this symbol, pressure potential, this one. And when you add solute and pressure potential together, that's how we calculate water potential. Okay, note that solute potential is just the potential energy of water if solutes are dissolved in it. If there's no solutes dissolved, the solute potential is 0. Okay, and it can then go towards the negative, but you can't have less than no solutes, and so we can't have a positive solute potential. It'll always be negative. Okay, and it will be more negative as it gets more concentrated. Okay, pressure potential, on the other hand, can be either positive or negative. The higher the pressure, the more potential energy water has. Pressure potential can be negative or positive. Okay, inside cells, pressure potentials are generally positive. Okay, with the exceptions of xylem vessels, where sap is being transported through transpirational pull under tension, that can have a positive pressure potential. All right, let's look at an example. Okay, here we have some distilled water. Okay, we have a plant cell. Okay, and we're going to be bathing it in a hypotonic solution, in this case, distilled water. Okay, note that the water, okay, is going to have a pressure potential of 0. And this is a little typo. This should say solute potential, just like each of these do. Okay, 0. It's distilled water. Now, immediately when the plant cell is put in, okay, it's going to have a pressure potential of 0, but has a solute potential of -2 because there's more solutes inside the plant, okay, than in the distilled water. We're going to see the water now immediately is going to begin to move in due to osmosis. So we're going to increase the pressure inside as there's more water moving in. So here we can see the pressure potential is going to keep increasing until this balances out. Okay, and so we have a water potential of 0 after some time of being in the water. Okay, and so that's just what we have here. If the bathing solution is pure water, where the water potential is 0, the plant tissue will reach that level. Okay, let's go. So very last thing, now the opposite, if we're bathing in hypertonic solutions with high concentrations, we're going to see the solute potential of the bathing solution is even more negative than that of the tissue. Okay, the pressure potential of the bathing solution is 0, but pressure potential in the plant cells is likely to initially be above 0 due to pressure being above atmospheric pressure. Okay, both the solute and pressure potentials give the cells a higher water potential than the bathing solution. And remember that pressure always goes from high to low. So there's going to be a net movement of water out of the tissue. Okay, this is going to reduce the pressure in cells. Okay, we're going to end up with flaccid cells due to water loss. And if this is going to continue until the solute concentrations have evened out. Okay, this is bad news for the plant cell. It's become plasmolized. The plasma membrane is pulled away from the cell walls. Okay, and this will continue until the concentration of the cytoplasm equals that of the bathing solution. So there's no more net movement of water at that time because the solute potential and pressure potential of the cells and the bathing solution are equal, and therefore their water potentials are equal. So it's just a different way of talking about the exact same concept of osmosis, but we're just using this term water potential, and water is just always going to go from high to low. All right, that's the end of that, and I wish you the best.